Hello everyone, welcome back to Medical Digest. So in today's video, let's talk about the topic clinical trials. It is one of the short notes as well as very important topic when it comes to any kind of entrance examination. It is basically an important topic because it has got overlapping in pharmacology as well as in community medicine, that is preventive and social medicine. When it comes to clinical trials, basically it is consisting of major 4 phases, that is phase 1, phase 2, phase 3 and phase 4. But before discussing these 4 phases in detail, let's see some basics. So when it comes to basics, first let's talk about the preclinical trials. When it comes to preclinical trials, these trials are basically done in animals because we cannot test any kind of drug as soon as it is being formulated directly on the human beings. It is not ethically possible, and it is against the guidelines of the human safety. So what we do is once the drug is formulated, it has been produced, we test it on animals. But also in animals there are certain guidelines. We cannot test a drug on any kind of animal of our choice. There are certain guidelines of the various committees. So the preclinical trials comes under the committee of CPCSEA, that is the Committee for the Purpose and Control of Supervision of the Experiments on Animals. These kind of committees and various other committees worldwide give us a certain guideline so that we can ethically practice the drug trials on the animals and study their effects properly. Okay, so once the drug is through it has got approved the preclinical trials, that okay, the drug has been introduced in animal and it has showed to have some efficacy. For example, we want to test any kind of hypertensive drug, so there are ways where we can make that animal hypertensive. There are many ways. So once we create hypertension in that animal and test that drug, and it has found that it is lowering the blood pressure of that animal, so this drug has been found to be effective in that animal, right? So we can say that so this drug has been found to be effective and has passed the preclinical trial which is done on animals. So now after this, the drug is ready to enter into the clinical trials which is done on human beings. Okay, now the clinical trials involved the drug trials which are done on human beings. So it is done according to the GCP guidelines, that is Good Clinical Practice Guidelines. And the licensing authority to do such kind of clinical trials around the world is US FDA, and in India it is DCGI, that is Drug Controller General of India. And this DCGI comes under CDSCO, that is Central Drug Standard Control Organization. So what we need to do, we need to write a letter to the licensing authority. This letter is called as Investigational New Drug Application, that is INDA. And after the approval from the licensing authority, the clinical trials start. So as you can see here, phase 0, but in order to understand the phase 0, first we need to understand what are the different first 4 phases. Like phase 0 is obviously before phase 1, but if you understand phase 1, phase 2, phase 3 and phase 4 properly, you will be able to understand phase 0 as well. So when it comes to phase 1, let's talk about phase 1 in detail now. Phase 1 usually involves healthy human volunteers, but when it comes to exceptions, it is mentioned as here, suppose there is a fatal disease like cancer in the end stage and someone has discovered a new anti-cancer drug, and that drug has been shown to have toxicity in animals. So these drug in preclinical trials already has been found to be toxic. So in order to introduce this drug in the human trials, we won't select any healthy human volunteer. We won't give any toxic drug to a healthy human volunteer, right? Because it is against ethics. So what we will do, instead of choosing healthy volunteers, we would prefer an end stage or terminally ill cancer patient, so that we can understand whether the drug works on this works on this patient properly or not. Okay, so generally in phase 1 it is done with healthy human volunteers, but when it comes to be doing a drug trial which is found to be toxic in a preclinical trial, and it is basically given to the patients who are terminally ill, so instead of choosing healthy volunteers, we will directly give this drug to a terminally ill patient. Now what this phase can do, it basically cannot test the efficacy of the drug. Why? Because in healthy human volunteers there is no symptom, so if we are giving the drug, there won't be any effect. If we have fever and we take antipyretic, and this antipyretic lowers down our fever, then this shows that the drug is efficacious. But if we are not having any fever and if we take an antipyretic, what effect will it do to our body? It will only maintain the normal body temperature. So basically we cannot test the efficacy in the phase 1 trial, but we can definitely test the safety of the drug. Why? Because in healthy human volunteers, if the drug is toxic, it will show some side effects, but if the drug is non-toxic, it will obviously won't have any kind of effects in the body. But what is the main aim? What is the main aim of the first phase of the clinical trials? It is to know the maximum tolerable dose. Like if we take again any antipyretic drug, and if we give 100 mg dose, nothing happens. Okay, we do all the tests of the human body in that healthy volunteer, and we send him home. Again we do the same thing with the 200 milligram of this drug, again on another person, and we do all the tests to see any kind of adverse effects, and we send him home. This we do until we get the maximum tolerable dose possible. We get the maximum tolerable dose. For example, we reach up to 1000 milligram, and 1000 milligram from we give 1000 milligrams to the healthy volunteer, and now it is showing that this patient has developed some kind of toxic effects on his body. So we come to know that okay, this drug can be given maximum up to 1000 milligrams. Alright, so the main aim of phase 0 trials is to know the maximum tolerable dose. After that, what kind of dosing patterns we can introduce in the market, for example like 100 milligrams, 150 milligrams or micrograms, like what kind of gramming should be done in the drug is decided in the phase 0 itself. Also we also study the pharmacokinetics to some extent in the phase 0, but before we properly understand it, but pharmacokinetic study is already done in the phase 0 if it is done. All right, and after that we also test the safety and the non-toxicity of the drug. So the first phase in human beings is the phase 1. It is asked that what is the first phase in human beings? It is the phase 1. Why? Because before this it is directly the preclinical trials which is done on animals, right? Now coming towards the phase 2. It has got two parts, part A and part B. So what is part A? It is called as proof of concept, and the phase 2 starts from patients. Now what we have done in phase 1, it is healthy human volunteers, but in phase 2 we have patients. So what is the first phase? What is the first phase in which patients are used as candidates? So it is the phase 2. When it comes to part A, it is the proof of the concept. So what is done here? It also tests the safety and the efficacy of the drug. But now here the dosing is given, and since the patients are used, for example, when it comes to antipyretic fever, we will select some certain fever patients, we will introduce the drug into the fever patients, and we will see whether the drug is having any effect on the fever or not. So for the first time, the efficacy of the drug is known in the phase 2. Now when it comes to part B, we start ranging the doses, like what kind of dose range should be preferred in different kinds of population. So this is the trial with multiple population with multiple dose range. So it tests the safety as well as the efficacy of the drug, and the group of people which we select is around 100 to 200 people, and it is usually unicentric. For example, people from the same source, example, patients from the single hospital, we take the patients from the OPD, send them to the ward, we introduce this drug and see whether the drug is efficacious in these fever patients or not. Okay, so what is the phase where maximum drug failure is seen? It is the phase 2. Why? Because in phase 1 we are not testing the efficacy of the drug. It is the phase 2 where first time the drug efficacy comes into picture. So here in this phase only the drug may either pass the test or may fail the test. So maximum drug failure is seen in phase 2. Also the first phase which is used in patients, where the patients are used, it is also phase 2. Also the phase in which we come to know about the efficacy of the drug, this is also phase 2. Now coming towards the phase 3. Okay, phase 3 is also done in patients. It is also called as pivotal studies, and it also tests the safety and the efficacy of the drug. But the group of people which we take is a large number of people, like around 5,000, and multicentric group of people is taken. Why? Because we need to see whether this drug which has passed the phase 2 trial is efficacious in other gene pool as well, in other type of people as well, because in phase 2 we have selected the group from a certain one source. Now we are testing this drug in different kind of people. So the source of the people or the source of the patients is multicentric, that is multicentric, so that the trial is on the people of different areas, states or countries. And also in this phase we will see that whether the new drug or the new treatment is good as compared to the existing drug available in the market. For example, when it comes to paracetamol, we know that paracetamol is a very good antipyretic. So if I have created a new drug called as drug A, and I want to see whether it is superior than paracetamol, then I will see this in the phase 3 trials, but I won't be seeing it in the phase 2 trials. Why? Because first I need to see whether the drug is working or not. If it is not working, if it is not bringing the fever down, what is the need to compare it with even paracetamol? Okay, so first we need to see whether the drug is being effective. Now what we do is we see whether the drug is being effective on different kind of people, as well as we will now compare this drug to be superior or inferior or as same efficacy or having same efficacy with the existing drug in the market. So with the example, obviously paracetamol, we will confirm its efficacy. So what is the phase which will confirm the efficacy of the drug? So this is the phase 3. Confirmation of efficacy of the drug is done in the phase 3. So what we do now, once, so I have invented or formulated the drug A, and I have found until the phase 3 that it is superior to the drug paracetamol, and yes, I have succeeded in my trials. Now what we do a letter, we write a letter again to the licensing authority. Now this letter is the NDA. The previous letter was INDA, but now what is the difference? This is the NDA. So NDA is the new drug application. So once it is approved by the licensing authority, my drug is launched in the market. So what is the phase where the new drug is launched in the market? After the completion of the phase 3, the new drug is launched in the market. And also, in which phase do we do randomized control trial? In the phase 3. Why? Because we are comparing the new drug with the old drug. So there is control as well as comparison group. There is a control comparison group, okay, which is given paracetamol. The drug trials are done in a fashion where we compare the drug with no drug, we compare a drug with placebo. For example, we comparison are done like new drug versus no drug, new drug versus placebo, and new drug versus existing drug. So this kind of studies is shown in the randomized control trial, and this is done in the phase 3. Now coming towards, and also the most important phase. Why? The most important phase of this trial is of the clinical trial is phase 3. Why? Because once a drug has been cleared or it has passed the phase 3 trial, we can obviously write an application and get the authorization to launch our drug in the market. So this is obviously the most important phase of the clinical trial, that is the phase 3. When it comes to phase 4, again it is done in patients. And now after the drug has been launched in the market, we will study it afterwards, after it is launched in the market. So it is also called as post-marketing studies or post-marketing surveillance. Okay, so why there is a need to study the drug once it has already cleared so many phases and it has been approved and authorized? Because some adverse effect development of the drugs take time, and these drug effects are called as rare side effects. For example, it is shown in 1 people out of 20,000 people, and also certain drugs take a very long time to show its adverse effects. So these are also known as long-term side effects of the drugs. So we cannot obviously spend a lot of time to know the rare side effects and long-term side effects before the phase 4, because we cannot spend our entire lives doing research only. We need to see, we need the person who is investing in a development of a drug also needs some kind of profit, right? So the drug will be launched once it is proven that it is efficacious as well as it has safety, it is safe. So for after that, the post marketing surveillance is taken place under phase 4, and we will now study the rare as well as long term side effects of the drugs. So as it is taken for a very long time, it is the longest trial. The phase 4 is the longest trial. It is actually lifelong, but it should minimum be up to 10 to 25 years. Also, the post marketing surveillance is done in phase 4, and the drug is being tested on maximum number of patients, because now the drug is launched in market, and once it is prescribed, it will definitely reach maximum people possible. So the phase which recruits maximum number of patients is obviously phase 4. Now when it comes to the blinding, okay, when it comes to blinding, so as we know that there are different kinds of blinding: single blinding, double blinding, and as well as triple blinding. It is another topic, but it is the blinding is basically done to remove the bias. The blinding is basically done to remove the bias. For example, the subject bias, which is done by the patient, the investigator bias, which is done by the doctor, as well as the analyzer bias, which is done by the analyzer of the study. So basically when it comes to single blinding, it is done in the phase 2. So what is single blinding? Single blinding is the patient is not knowing what treatment is given to him, but the doctor knows what kind of treatment is given to him. So this is done in the phase 2. When it comes to double blinding, it is done in phase 3. Why we want that doctor should also not know what is given to the patient? Because now the analysis and the randomized control trial begins in the phase 3, and the doctor is the basic investigator, and he will ask the questions regarding the drugs. So if he knows that okay, this patient has been given a new drug and the other patient has been given the older drug, so there is some kind of bias which is created in the investigator's mind, so that he may mark the findings according to his own conclusions. So we are not supposed to do that. We want it to be completely neutral. So the double blinding studies are done in the phase 3, but the single blinding studies are done in the phase 2. But when it comes to phase 1, there is no need for blinding, because healthy human volunteers are selected, as well as there is no kind of efficacy which is being tested. So the drug is given under open label. So open label studies are done in phase 1. So this completes our all the phase 4. Phase 1 done in healthy humans, phase 2, 3 and 4 done in patients. Until this, we get a comprehensive understanding of all the clinical trial phases. Now coming towards the phase 0 trial, which is called as microdosing. So what are the drawbacks of starting clinical trials directly from the phase 1? If we directly start the clinical trials from the phase 1, what will happen? Suppose while doing a phase 1 trial on 100 healthy volunteers, 3 of them died due to a complication or adverse drug reaction. So this creates a poor design which killed unnecessarily 3 healthy people, just in order to test the safety and the non-toxicity of the drug. Also, the drug given may not show the desired amount of concentration and the effect on the targeted organ. Involving many people to take such drug will be waste of time as well as money. So what we can do to solve these kind of hindrances and problems? So we will introduce a phase 0 before phase 1. It is also basically done in healthy human volunteers, but what is the difference before phase with phase 1? In phase 0, we give the dose, but to a very small number of people, like 1 to 3, and also in very little dose. So the drug is given to a very small number of people, that is 1 to 3. Also the drug is given in a very less quantity. The maximum dose limit which we keep is usually 100 micrograms. It is 1/10 to 1/100th of the human dose given. And also important, very important, a radioligand is attached to the drug, so that we can study all the kind of micro level activities which this drug does in the body, and also to study the pharmacokinetics of this drug. So what happens in phase 0? It does not test the efficacy as well as safety. Why? Because in small dose it will not cause any change. It will not cause any change in the body, so it will not show any kind of effect in the body. Also, when it comes to safety, small dose being given, it will not have any kind of side effects. But the drug properties will be studied extensively, and unnecessary least effective drugs will be cancelled, and only effective drugs will be allowed to go further into the following trials of the phase 1, phase 2, phase 3 and phase 4. So this discussion completes our topic, that is clinical trials. I have tried my level best to explain this topic. Hope you like it, and please do share your views. Also do share this video with maximum of your friends. Until then, see you in my next video. Take care. Bye bye.